Okay, let's jump right into this explainer. Today, we're unpacking the molecular biology of HIV. And honestly, we're going to treat this like a true crime story, just, at the cellular level. Think of this virus as a highly specialized intruder, pulling off a completely brilliant microscopic heist inside the human body. It's fascinating, it's totally devious, and understanding exactly how it operates is really the only way we've figured out how to stop it. So here's the blueprint for our heist. We'll start with HIV versus AIDS, then look at the anatomy of the virus. After that, we'll cover the microscopic break-in, hijacking and making copies, and finally the fall of immunity. Section 1. HIV versus AIDS. Understanding the basics. Now, before we actually analyze the break-in, we need to correctly identify our culprit. And really, we need to clear up a massive, common misconception right out of the gate. The absolute most crucial point to understand here is that AIDS, acquired immunodeficiency syndrome, is the devastating end-stage condition. It's the syndrome itself. It happens when the body's immune system is just heavily compromised by the actual intruder, which is the Human Immunodeficiency Virus, or HIV. So this clearly separates the two for us. On the one hand, we have HIV, the virus itself. That's the infectious pathogen getting transmitted through specific bodily fluids. On the other hand, we have AIDS, the condition, and that's the collection of symptoms and opportunistic illnesses that emerge when the immune system is severely damaged. So having HIV doesn't automatically mean a person has AIDS. It just means the intruder is inside the building and has breached the perimeter. Moving right along to Section 2, Anatomy of the Virus, Structure of HIV. Let's take a close look at the actual physical tools and disguises our microscopic intruder uses to pull off this biological heist. Now, HIV is an enveloped retrovirus, specifically a lentivirus, and man, it comes fully equipped for infiltration. It features this lipid envelope, which is actually a host-derived membrane. It literally wears a disguise made from the very cells it previously attacked. Protruding right out of this disguise are the GP120 and GP41 glycoproteins, and these basically act like grappling hooks. Deep inside, there's a protective protein armor called the capsid, and hidden inside that vault, the genome. That's two identical, single-stranded RNA molecules packed tightly alongside specialized enzyme tools like reverse transcriptase, integrase, and protease that are designed specifically to pick cellular locks. And what's truly mind-blowing here is how the virus's genome acts as the exact, specific blueprint coding for these infiltration tools. It uses three major structural genes. The GAG gene acts as the blueprint for the capsid and matrix proteins, so the armor. The POL gene encodes those essential enzyme tools to pick the locks. And the NV gene encodes the envelope proteins GP120 and GP41, the grappling hooks. It is a highly efficient, incredibly compact blueprint for a biological break-in. All right, Section 3, the microscopic break-in, HIV entry to host cells. With our tools identified, let's watch the intruder actively target and breach the body's primary defenders, the CD4-positive T lymphocytes, macrophages, and dendritic cells. This sequence brilliantly illustrates the intrusion, mapping it out in 4 distinct steps. First up, attachment. The viral GP120 binds directly to the host's CD4 receptor. Second is co-receptor binding, where the virus binds to a secondary lock, usually CCR5 or CXCR4. Third, fusion. Here, the GP41 protein acts up, physically mediating the merging of the viral and cellular membranes. And finally, step 4, entry. The membranes open wide, and the virus dumps its payload, its viral RNA and enzymes, straight into the cell's cytoplasm. The alarm hasn't even triggered, but the intruder is officially in. That brings us to Section 4, hijacking and making copies, reverse transcription. Because once it's inside, the intruder must overwrite the cell's operating system to illegally manufacture copies of itself. Notice the stark contrast in how this operates? Normal cellular transcription reads DNA to make RNA, right? But here, the retrovirus runs the whole process backward. Using its reverse transcriptase enzyme, it synthesizes complementary DNA straight from its RNA payload, while the original viral RNA gets degraded. Then, it synthesizes a second DNA strand, resulting in double-stranded viral DNA. It has literally rewritten itself into the language of the host cell. But here's the catch. This sloppy, backward translation process is highly error-prone. And this isn't just a quirky biology fun fact. It's the exact reason HIV mutates so rapidly, evades the immune system so efficiently, and develops such severe drug resistance. It is constantly, relentlessly shifting its molecular disguise.
Let's look at how this builds into the final phase of the replication cycle. First, we have integration. This is where the integrase enzyme literally hides the new viral DNA inside the host's own chromosomes, acting as a latent sleeper agent called a provirus. Next is expression. When activated, the host cell's own machinery unknowingly synthesizes viral mRNA and proteins. Then comes assembly, where the new viral RNA and proteins gather up at the plasma membrane. And finally, budding and maturation. Immature virions escape the cell, and the viral protease steps in to finalize the structure, forming mature, highly infectious attackers ready to go out and repeat the whole process.
Moving on to Section 5, the fall of immunity, progressive CD4 decline. Because the success of this microscopic heist at the cellular level, it leads to a catastrophic collapse of the entire human immune system over time. Now, while the virus is absolutely not spread by casual contact like hugging or sharing food, its initial transmission at day 0 via specific bodily fluids triggers a truly relentless process. You get this prolonged period of continuous viral replication, which causes gradual immune dysregulation and apoptosis, or cell death of infected cells. Without medical intervention, this triggers a slow 8 to 10-year systemic destruction of the immune system. And that culminates in the final stage, the onset of AIDS and an intense susceptibility to opportunistic infections and cancers. Take a good look at this specific number, 200. See, a normal CD4 count in a healthy individual sits somewhere between 500 and 1500 cells per cubic millimeter. But dropping below this critical threshold of 200, that is the point where the immune system officially collapses. Literally, this is the clinical definition of transitioning into AIDS. And the mechanics behind this collapse are completely multifaceted. The sheer, overwhelming volume of this microscopic invasion triggers massive cell death through direct destruction, apoptosis, and, get this, even the immune system mistakenly killing its own targeted cells. When you combine all that with reduced helper T-cell function and defective macrophage activity, you end up with widespread immune dysregulation. It leaves the biological vault completely unguarded against everyday, otherwise harmless diseases. But we have Section 6, the counterattack, antiretroviral therapy. There is a powerful scientific response here. While there is currently no cure, understanding the exact molecular biology of this heist gives us the perfect blueprint to fight back and manage the condition highly, highly effectively. Antiretroviral therapy, or ART, uses targeted drugs to block every single specific phase of the viral life cycle we just explored. Entry inhibitors block the grappling hooks and fusion. Reverse transcriptase inhibitors jam up that backward coding process. Integrase inhibitors put a stop to the DNA integration, and protease inhibitors prevent the virus from maturing into an infectious state. By taking a specific combination of these daily, we can suppress the virus to entirely undetectable levels. This allows the immune system to recover and halts the progression to AIDS completely. A real game changer. Which brings us to an ultimate, lingering question. Given that this intruder actually weaves its blueprints directly into our own cellular vaults as a latent sleeper agent, what will it take to finally, completely eradicate this virus from the human body? Understanding the heist was our first victory, but evicting the sleeper agent? Well, that's our next great frontier.